In this video, we're going to finish the topics of chapter 7. The first thing that we're going to talk about is the hydrogenation reaction that can be performed on molecules that contain fatty acids. Hydrogenation reaction, which specifically I'm going to discuss commercial hydrogenation, is the reaction in which hydrogen, it is added to a vegetable oil that contains unsaturated fatty acids and is going to turn it into a molecule that now is a saturated one. So if we look at the bottom and we look at the structure for the molecules that we have in the example, let's notice that the molecule that overall looks at or is described in oils, or is present in oils, I should say, is called a triacylglycerol. Triacyl comes from the idea that we have these C double bond O, single bond oxygen motifs. Glycerol means this area of the molecule that is in purple is where glycerol is. And maybe you haven't heard the word triacylglycerol, but maybe you have heard the word triglyceride, which is one of the biomolecules that exist in our body because it's how we store fat, is in this molecule. Now, when it comes to a triacylglycerol or triglyceride, we can see that this molecule has 1, 2, 3 molecules of fatty acids that can be stored in this molecule. Now, the example that we have here specifically refers to a triacylglycerol that is fully composed of fatty acids that are unsaturated. And we know that they're unsaturated because each of the fatty acids in the molecule are going to have a carbon-carbon double bond. These carbon-carbon double bonds, as we observed earlier, tend to have a cis conformation in terms of the double bond. When hydrogenation of these products, which because of the presence of unsaturated fatty acids tend to be liquid oils, and hydrogenation happens, then that hydrogenation, what is going to do is that is going to place hydrogens on each of the carbons that used to be as part of the double bond. So when we look closely at the molecule, we can observe that specifically the molecule in the beginning, what we started with, has 7 CH2s here and 7 CH2s here. So overall, it had 14. Upon hydrogenation, because each of these CH groups turn into a CH2, that's why we observed here that there's a total of 16. Because the initial 14 are present, 2 more CH2s are generated. That's why we see that this changes to CH2 16. In addition, let's notice that because each of the fatty acids remaining in the molecule, the one in the middle and the one in the last position, also have one double bond, then we can see overall that each one of those turns into CH2s, and that's why we see the number CH216 in the product. It is important to note that when commercial hydrogenation happens, it happens only partially. So that's why when you buy products like margarine or shortening, you will see that it says partially hydrogenated oils is what's present in it. The reason why we only see partial hydrogenation is because these products tend to have the state of matter of a soft semi-solid fat. The benefit of partial hydrogenation is that by introducing some of the molecules, because partial hydrogenation means that only a certain subset of molecules get hydrogenated, not every single one. The ones that are saturated now in the product and when they are placed in the food product, understand that the saturated products are going to have higher melting points. Now, there is some problems with this reaction. It's not a perfect reaction because along with the saturated product that is desired, there's also a byproduct that can be produced. So as I mentioned a few moments ago, when it comes to what we start with, this molecule actually is a cis fat. In this reaction, you can actually have a side reaction in which that cis oil can be converted into a trans oil. So those alkenes can go from cis to trans. Now, this is actually not favorable. The reason why trans fats are actually linked to diseases is because in, for example, our bodies, we don't have the biomolecules in order to process those trans fats. So what happens overall is that sometimes they get accumulated. So that's why we think of trans fat not being the best for you. Lastly, we are going to talk about the concepts of attractive forces and cell membranes. In the beginning, when we started talking about lipids, we saw that lipids have different compositions. We saw the two large families, fatty acids and steroids. And when we look specifically at the structures that are present in animal cells, including the cells in our body, it's important to note that these lipids that we were just talking about are present in many areas of the cell. Cell membranes specifically, which are responsible for separating the cellular content of inside the cell from the external environment, have lipids in them. Cell membranes specifically consist of what is called a lipid bilayer. So what this means is that we have two layers of lipid present in the molecule. Now, when we look specifically at that lipid bilayer, we can see that this bilayer is made from phospholipids. You can go back in the slides and look at the structure of a phospholipid. These phospholipids overall have two portions of fatty acids, which are represented by the two orange scales that you see in the structure. So right there. And then it has a polar head, which that's where we have the phosphate and an amino alcohol. And that's represented by these green circles that you see all over. Now, we can see that overall this lipid bilayer, it is arranged such that the phosphate group and the amino alcohol that is represented by those green balls are going to be facing inside of the cell and outside the cell. And the phospholipids are going to meet specifically by the tails. Because these tails tend to be nonpolar, that's why they're interacting with one another. In addition, cell membranes have another important lipid, which is cholesterol. Cholesterol is actually present here. We can observe that these areas of yellow molecules is what cholesterol is representing. And we can observe that this is in place within the lipid bilayer to help its structure. In addition, we have other components that include carbohydrate chains, which we already talked about carbohydrates. And in later chapters, we will be talking about other important biomolecules, including proteins that are also present here. Now, whenever we talk about cell membranes and attractive forces, we need to understand that because these structures have different chemical features, they are going to be interacting with one another via different attractive forces. In terms of the content that is present in the cell membrane, this takes us to at least discuss what is the fluid mosaic model to define why is it that these cell membranes have the structural components that they possess. So in this fluid mosaic model, which is what explains the components of a cell membrane, the reason why this is called fluid mosaic model, it's from the idea that the mosaic portion of the model, let me use my highlighter too, it's actually represented by the variety of components that are there. We have unsaturated fatty acids. We have carbohydrates. We have proteins. We have cholesterol. So there's different things that are present in the molecule. Now, the fluidity of the fluid mosaic model, it actually is defined by the idea that in these components that make a membrane, the unsaturated fatty acids, because we saw before how unsaturated fatty acids have more like a liquid consistency, and since they are the main fatty acid that we see in the phospholipids that are part of the structure for the membranes, then that gives the membrane a more fluid-like structure than rigid. But in a way that makes sense because these cells are going to be undergoing changes and moving around and are going to be intaking molecules and actually transporting molecules throughout the cell. So they need this flexibility. In order to understand further the idea of attractive forces and membranes, we need to talk about the concept of transporting substances in and out of the cell. So when we look at the cell membrane, let's establish that specifically the area of the molecule that is represented by the different green portions, the green spheres or balls, this is actually going to be the polar portion of the cell membrane. So it has two polar portions. Now, this area where we see these tails, this is going to be non-polar. So when it comes to the attractive forces that are making those tails meet each other, it's mostly going to be dispersion forces. And when it comes to what are the attractive forces that are going to be encountered inside of the cell or outside of the cell, they are going to be dipole-dipole attractions, or in the case of ions, there may be some ions that could be attracted to those portions, but it's mostly going to be maybe some dipole-dipole, possibly some hydrogen bonding. Now, how does this affect how substances come in and out of the cell? Well, when it comes to transporting substances, which is very important for the maintenance of the system or biological systems, there are going to be at least 3 different ways in which chemical compounds can come in and out of the cell, starting with diffusion. Diffusion, which is also known as passive transport, is going to allow certain particles or molecules to move from a higher concentration gradient to a lower concentration gradient. So when we look at this, molecules that can diffuse through the cell include small nonpolar molecules. So for example, O2 or CO2, which means molecular oxygen or carbon dioxide gas, can pass through a cell via diffusion. Another molecule that is interesting and is able to do this even though it's polar, it will be water. So some water molecules can actually come in inside of the cell via diffusion. Now, what happens to molecules that cannot come in and out of the cell by diffusion? Well, they have what is called facilitated transport. And facilitated transport is going to be that middle illustration that we have on this figure. Facilitated transport basically says that there is a protein channel, and proteins are just a different type of biomolecule. There is a protein channel that is going to increase the rate of diffusion. So in a facilitated transport type of transport, that channel, as you can see, is a protein that kind of like creates a tunnel that is specific for molecules come inside of the cell. Again, via facilitated diffusion, because this protein creates that channel at the membrane that allows particular molecules to enter. One of the examples of molecules that can actually come through the cell through facilitated transport also includes water. And then the channel that allows water to come into the cell is going to be called an aquaporin. And another molecule that can actually come in and out of the cell through facilitated transport includes glucose, which is a sugar. Lastly, if a substance cannot get into and out of the cell via diffusion or facilitated transport, we have a third mode of transport, which is called active transport. In active transport, we are also going to utilize a channel, which is a protein that is going to allow the molecules to get inside and out of the cell. But specifically, our active transports is due to the use of energy. So this is going to happen specifically because energy is going to be spent in order for these molecules to be transported. So one of the main molecules that we see that is going to be transported via active transport include ions. Ions like sodium, potassium, and calcium use these type of transport systems in order to get in and out of the cell. So it is important to note that attractive forces are mediating these interactions overall because of how the environment of the cell has to be maintained.